and I will be talking, oh, it's not, oh. Yeah, so far so good. Okay, great. I will be talking about NetworkAssessor. This is a web tool for cancer precision research in medicine under development at the Riva Lab at the Icahn School of Medicine at Mount Sinai. I'm going to be talking about the who, what, where, when, why, and how of this tool. So the very first question, of course, is why? Why am I here, and why bother building this tool at all? There are excellent tools, online tools, for gene set enrichment analysis, namely Enricher, David. Reactome has its own tool. And what this allows you to do is to take a gene signature of a given number of genes. And if you have, let's say, M genes overlapping with pathway A and M genes overlapping with pathway B, thanks to gene set enrichment, you know which pathway is more significant. However, there still remains a question, what if M equals 0? What if you have no overlap? Does that mean that these pathways aren't significantly related to your gene set of interest? We think that by leveraging existing protein-protein interaction databases, we can tell whether or not these pathways are still of biological interest to your query gene set. So if you have a gene signature of N genes and they form M edges to pathway A and M edges to pathway B according to existing PPI databases, we can assess which is more significant. And we can do this with NetworkAssessor. So we've developed this method to assess the significance of edges between an experimentally derived gene set and a pathway by leveraging existing PPI databases, much in the same way that gene set enrichment analysis allows you to leverage existing gene collections such as pathway databases and gene ontology annotations. So how do we do this? First, we take a randomly generated set of size n, a gene set of size n. So for example, in this case, we'll take a look at a set of 3 genes. They form their own subnetwork according to these existing PPI databases. A given pathway will also perform its own subnetwork. And we simply count the number of edges between our gene set, our randomly generated gene set, and the pathway. And we do this quite a few times. We'll just generate random gene sets over and over and over again. And this will, right now what we have is up to 100,000 times for gene sets of sizes 1 to 200 genes. And we'll do this for each pathway. So we end up with pathway-specific distributions for a given number of genes. And based on this distribution, we can essentially assess the significance of a gene set of a certain size to a pathway, to a given major cancer pathway. And I just wanted to show this slide to show you that we've done this for lots of pathways, about 30 major cancer pathways with lots of different gene sets. And as you can see, as the gene sets get larger, you get this really nice normal distribution here. So what exactly is NetworkAssessor? It's a web app for visualizing these gene interaction networks in the context of molecular pathways of interest. Of course, in addition to the visualization, you get the analysis, and you will have the major cancer pathways returned ranked according to the most significant to the least significant. The pathways of interest right now are cancer pathways annotated by My Cancer Genome, and eventually we hope to integrate KEGG pathways and reactome pathways also. The underlying premise is that genes of interest that make numerous interactions with genes of major cancer pathways likely contribute to cancer progress. By this we mean tumor invasion, metastatic invasion, tumor growth, etc. So now that we know what it is, what is something that you can use it for? Here we did a little experiment where we are looking for a KEGG pathway with no genes in common with any of the major cancer pathways, but that interacts strongly with at least one major cancer pathway. If we find this, it suggests that genes of this pathway plays a role in cancer that couldn't have been found with gene set enrichment analysis alone. And so how do we do this? When you're looking at gene set enrichment analysis, what you're essentially looking for is the intersection between a given input. So for example, in this case, a KEGG pathway and cancer pathway genes. This is what I call an intersection-rich pathway if it's significantly enriched. What we're looking for are essentially intersection-poor pathways, a KEGG pathway that has genes that don't overlap with any of the major cancer pathways and therefore is not very clearly associated with cancer. And then using this intersection-poor pathway, we wanted to see if it interacts with how many major cancer pathways it interacts with. If it doesn't interact with very many, we consider that an interaction-poor pathway. But if this pathway interacts with lots of major cancer pathways, we consider this an interaction-rich pathway. So What we found was that the KEGG proteasome pathway is an intersection-poor pathway, meaning the genes of the KEGG proteasome pathway don't overlap with any of the cancer pathway genes. However, it does form significant relationships with many of the major cancer pathways. And so what happens when we put these genes into NetworkAssessor, the genes of this pathway into NetworkAssessor? So we're going to go ahead and paste those and click Analyze. And what you end up with is a network visualization with a ranking, oh, I'm sorry, it's quite blurry, with a ranking of the major cancer pathways according to their significance. As you see, what you end up with here is this, right now this is the network according to BioGrid, so the pathway is really highly interconnected. And in particular, it's highly associated with protein degradation ubiquitination pathway. And the MDM2 gene seems to be the primary contributor to the high number of edges formed between this pathway and the pathway of the KEGG proteasome. I also wanted to show that you can review with our server higher degrees of connection between the pathway and the query set. Blue edges indicate pathway-to-pathway connections, and red edges indicate a connection to at least one query gene. It's very interactive. I'm using Cytoscape.js here. And there are other little features. So for example, you can use, we have a handy visualization tool where you can differentiate between the pathways and tailor the visualization to how you want it to look. There's a legend. You can move the nodes around. Let's see here. Yeah, you can toggle the legend on and off. You can add on as many significant pathways as you'd like and slowly build up your network according to what you think is the most biologically significant. Okay. I'm just showing how interactive this is. It's very pretty, if I do say so myself. And then, and it's live actually on networkassessor.net. And you can see here also you can see the list of the pathway genes, the number of edges that are formed, and the significance that we calculated. You can toggle between multiple PPI databases, including HPRD, and you can see the topology of separation of multiple degrees of separation between the pathway and your query set, and you can see the entire pathway if you wanted. So what did we end up finding with this particular exercise was, I said before, the KEGG proteasome pathway is intersection poor and interaction rich. In particular, it interacts strongly with the MDM2 gene of the protein degradation ubiquitination pathway. So therefore, we propose that the KEGG proteasome pathway plays a major role in cancer through its relationship to MDM2, something we found by looking at the enrichment in edges and not through the enrichment in genes. So something we found by looking through the edge space, essentially, and not in the nodes. And luckily for us, this result was actually confirmed in the literature, where we found that there is a major role of proteasome genes in cancer. For example, all the way back in 2005, they found that MDM2 promotes proteasome-dependent degradation of retinoblastoma protein. And then as recently as last year, they've had successful clinical trials using proteasome inhibitors in cancer therapy. And there are even more of such pathways in Reactome. We found about 200 pathways that you can start exploring in Reactome using this tool. Now the question is when? We look towards the future. Our next goals are, we're very much still in the prototype phase. The next goals are to continue development of the tool for research purposes, including assessing the significance of interactions with subnetworks formed by KEGG and Reactome pathways. I'd like to include a built-in pathway enrichment analysis. I'd like to contribute to more directed research by adding a feature of filtering input by gene ontology terms, and when it's annotated, to add directional interactions of the pathway genes. We'd like to develop the tool for personalized analysis of cancer profiles by adding cancer driver gene modules derived by TCGA, and adding interactive visualization features, including changing node features such as the size, shape, and color based on molecular features. Where can you find this? It's at networkassessor.net. It's totally open source, github.com/revoslab/networkassessor. We're in the process of releasing a RESTful API so that you can access this analysis programmatically. And since this is the technology track, I just wanted to give a nod to the technologies that I use. On the back end, I use a Python-based MVC framework, Django. Specifically, the library that I rely on most heavily is NetworkX. On the front end, I use a reactive front-end framework called Vue.js, and then I also use Cytoscape.js for the network visualization. And who do I thank for all of this? In addition to you guys, because I thanked you earlier. Boris Riva, who's here. This he made me be here, Hui Li, Dmitry Rikonov, and Eric Schatt. And thank you for having me.
Thank you very much, Anna. So we do have some time for comments, questions, thoughts.
Okay. Great.
Okay, well, thank you, Anna.
Thank you.
That was great. I think what we're going to do is we'll just wait